Class centered on a particular technology which is called phage display, and the easiest way to think about phage display is first to understand that phage is just a virus, but it's a virus that attacks bacteria, not us, so the students were safe. And the second thing is that because they're such simple things, we can co-opt their DNA, and in fact what gets generated are phage that are each a little bit different. So a single tube of phage actually has, you know, a billion or a trillion rather different virus particles that are in it, and you know, you can think about these like antibodies. So you know, you encounter a new disease, your body makes a new antibody, and you fight off the disease. And here we're doing that sort of at the bacteria and bacterial virus level. So why this is special is that in the case of a virus like a phage, what happens is that the part that might stick to a foreign material, or in our case, you know, material in an artwork, carries with it the instructions for its own formation. So once you identify a virus that sticks to, you know, a violin lacquer or a tortoiseshell, then you can take that virus and you can expand it, or rather make many copies of it at the detriment of all the others that didn't stick. So this was the core technology that we were trying to use. It's commonplace in the biomedical research community. It has had some enjoyment of use in the material engineering community. It has never been used in the art, I promise, has never been used in the art conservation community.
So we started off by taking the students to look at various objects in the art gallery and just talking about the object and how it was constructed and the kind of things that might have happened to it over time, and why we might want to ask particular questions like, you know, is this overpaint, or is this linseed, is it painted in linseed oil or walnut oil, for example? So it's quite difficult to distinguish them. And then, as well as taking on the gallery, we also took them to our conservation labs out at Hampden, and we were able to show them how we worked and the equipment we used and the kind of objects that we were working on. And another trip was also made to the Centre for Conservation and Preservation Research Labs out on West Campus, which opened in April 2013.
So really there were two halves to the class, and the first half we worked in the art gallery with the objects at the art gallery, and the point of this sort of introductory period of the class was to get the students starting to think like technical art historians, that is, to look at works of art as objects and to really sort of start to think about the questions that could be answered by knowing what the materials are of those objects.
In planning this course, we got very excited about planning the space, really. And so we paid a lot of attention to creating an environment for the students where they could really become a scientist, think in ways that scientists think, and in an environment that scientists generate ideas and share with each other, and also be surrounded by posters of art and artist materials. And so we created this lab space and really a curriculum that brings students full force through what it means to become part of a lab, part of a working scientific group. And so we have a lot of peer evaluation built in, and we had a lot of communication about projects between the students, and because they each had their own individual projects, we saw the excitement of that crosstalk between students develop, and that was great to see.
As a psychology major, it was really cool for me because I knew almost nothing about art history or about research in biology and chemistry. So it was really cool to sort of approach all of these topics all at once, and especially cool as someone who hasn't done lab research before to get exposure to the kind of thinking and the kind of process that goes into answering these kinds of questions. Eventually settled on the varnish on violins, and I was interested in this for a couple of reasons. So first, the varnish plays a really important role in how the violin and actually other instruments sound, and I also thought that a project involving varnish might be more applicable to a variety of types of art because varnish is used on furniture, it's used on harpsichords, it's used on violins, it's used on paintings. So I think there are a lot of applications for understanding the composition of varnish. And so looking more specifically at my project, I wanted to figure out the exact composition of a given varnish. And for violins in particular, it's interesting because there isn't a lot of information about what resins were used in a particular varnish, and they were often used in combination. So for my project, I decided to look at two varnishes, or two resins, that were often combined, one called sandarac resin, which is from tree, and then one called seedlac, which is from an insect.
Ever since I decided to become a history of art major, I've really focused on the decorative arts. So I wanted to come up with a project that would in some ways relate to my interest in the decorative arts. I floated around a few ideas. I really love wood, so I was trying to think of a project that could relate to wood. And eventually we settled on the idea to look at tortoiseshell veneer on frames. In particular, there's one frame in the art gallery's collection that we're not sure if it's tortoiseshell or not. And so that frame really got my attention, and we started this idea of thinking about, well, how do you identify tortoiseshell, and what are the implications of being able to identify tortoiseshell? Well, it's really important to identify tortoiseshell because, first of all, trading tortoiseshell is illegal. So if there could be a simple tool that could be used even at border crossings for people who are trying to traffic tortoiseshell, if the border control could check with my tool, that would be amazing. But also, it could be used in conservation to help conservators devise appropriate conservation techniques. Something really exciting about this project was that we were all encouraged to contact experts in the field that related to our areas of interest. So I met with 2 scientists at the Peabody who actually brought in a dead turtle that we got to get my samples from, and I got to talk to them about their experience in tortoiseshell identification and really learn from people who are doing it in the day-to-day. By the end of the time we spent in the lab, I actually had come up with a tool that could be used to distinguish between horn and tortoiseshell, something I never thought I would be able to do coming from an art historical background. And it's just really incredible that even experts in the field, such as the ones I spoke with at the Peabody, think that this tool is going to be valuable for them.